Hello, everyone, and welcome to the webinar, Unlocking the Code, the Era of Exomes and Genomes. My name is Kirsten Kelly. I'm a medical science liaison and part of the medical affairs team at GeneDx, and I will be moderating the webinar today. Before we get started, I wanted to point out a few housekeeping items. All attendees will be on mute, and we will have time for Q&A at the end of the presentation. And please enter any questions you have for the presenters in the question pane of the GoToWebinar control panel. We'll try to answer as many questions as we can in the time remaining. This live webinar has been approved by the National Society of Genetic Counselors for 0.1 CEUs as part of a webinar series. You must complete the webinar evaluation to obtain the CEUs. The evaluations will pop up at the completion of the webinar and will also be emailed out tomorrow to all attendees. Evaluations must be completed by April 3rd, 2025, and please be sure to include your NSGC ID number in the evaluation form. CEUs are sponsored by GeneDx, and they will be issued by NSGC when the series is complete. Also, this series has been reviewed by the Canadian Board of Genetic Counseling's Continuing Education Credits Committee, or CEC, and has been preliminarily approved for up to 4 CECs. We have several learning objectives for today's webinars, including summarizing the benefits and limitations of various genomic technologies, including panel testing, chromosomal microarray, exome sequencing, and genome sequencing. Describe exome and genome sequencing from sample to result. Review proband, duo, and trio analysis and the respective diagnostic yield differences. And finally, discuss exome and genome data reanalysis processes and their impact on diagnostic yield. It's my pleasure to introduce our speakers for today's webinar. First, we have Dr. Britt Johnson, who is a board-certified clinical molecular geneticist and clinical biochemical geneticist with more than 12 years of experience in medical affairs and clinical genomics. She's held various lab director and medical affairs roles with a focus on inborn errors of metabolism and inborn errors of immunity, supporting biopharma partnerships and sponsored testing programs, and providing medical support for platform technology initiatives related to variant detection and variant classification initiatives. And she's currently the head of medical affairs at GeneDx. We also have Dr. Maria J. Giansacoto, who is a physician trained in internal medicine, clinical genetics, and clinical molecular genetics. Her career has focused on new disease discovery using the power of genomic testing. She leads the team of gene disease validity curators at GeneDx and has collaborated in over 50 publications helping families with rare diseases find answers. And she is currently a director of clinical genetics at GeneDx. Here are the speakers' disclosures. And now I'd like to welcome Britt to help kick off the presentation.
Hi, everyone. Really excited to be here today on our first webinar at GeneDx in 2025 of this series. I think you're going to find that it's going to be a really foundational webinar, and our plan is to build upon this throughout the year with additional webinars in the series. So I'm going to kick it off and talk about some basics, and then I'll hand it over to Maria to go deeper on exome and genome sequencing. So with that being said, let's jump in. Next slide, please. So today I'm going to be talking about two of the major technologies that are used in the clinical lab. We have chromosomal microarray, or CMA, and next generation sequencing, also called NGS. So CMA tests for extra or missing pieces of chromosomes, which we call copy number variants, right? Since the result is a change in the number of copies of DNA that you have for the given segment. Additionally, CMA can also be used to identify regions of homozygosity, which we'll talk about a little bit more in the following slides. Now, next generation sequencing, on the other hand, is analyzing multiple fragments of DNA simultaneously to really determine what the actual DNA sequence is, and can also detect copy number variants as well. Multi-gene panels, exome sequencing, and genome sequencing all utilize next generation sequencing technology. Next slide. So first, let's talk about microarrays. The major microarrays that are used in the clinical lab today for chromosomal microarrays are what we call SNP arrays. Now, to perform a microarray, first what we do is isolate the genomic DNA from the patient's sample, and then we'll digest it with restriction enzymes to get smaller DNA fragments. And from there, we'll add adapters to the end of the DNA fragments, so then we can PCR amplify up these fragments and add a fluorescent label to the ends of the fragments. Now, these amplified fragments are then hybridized to an array, which is followed by laser scanning and computer processing of the data into a plot that displays the copy number and the genotype information. Now, the SNPs, right, and this is why they're called SNP arrays, because there's probes, and we'll talk about that in the next slide, but for the SNPs, we refer to them as A or B alleles on an array. So in the normal diploid state, right, when you have two copies, the genotypes you can get at these SNP sites are AA, BB, or the heterozygous AB. So in this example, you can see there's a deletion in a segment of chromosome 12, which results in the loss of one of these alleles and shows a corresponding loss of heterozygosity. And that kind of that bottom green line, you can see that that the section under there is a little bit lighter and has less green, because that's in that region, you only have a single A or B allele in this region. Next slide. So there are multiple array platforms out there that are used, but these SNP arrays typically use somewhere between 1 million and 3 million probes throughout the genome. Now, SNP arrays actually have two types of probes that are used. We have the non-polymorphic probes, which are used for the copy number variant detection. And arrays can detect typically, greater than 25 kilobases, size deletions and greater, and on duplications, typically it's about 50 kb and greater size of those duplications, but this can vary slightly from array platform to array platform, or also depends a little bit on the reporting policies of the lab. Now, the second type of the probes that we see on these arrays are the SNP probes, right? That's where they get their name. And these are really used to call regions of homozygosity. And those regions, when we see those, right, it's a loss of those heterozygous calls that can be due to uniparental disomy, which is when the two chromosomes in that region were inherited from a single parent and none inherited from the other parent. Regions of homozygosity can also be seen when there's consanguinity or relatedness in the family. Now, these regions of homozygosity and UPD typically can be called at a minimum resolution in the range of 3 megabases to maybe 10 megabases, depending on the array platform used and the policies of the lab. Next slide. So now let's take a look at 3 examples of the types of variants we can find on array. The right panels here are zoomed in images of the variant regions in the black boxes on the left-hand side images, right? So you can see at the deletion example here that there's a reduction in the fluorescence intensity at the top of the image. That's that top kind of purple line, and you can see the dip. And if you look lower in that image, you see that there's 3 horizontal, like purple lines at the bottom. And those represent the 3 different genotypes for the SNPs that are seen in that region, because you have AA, AB, and the BB calls at each of the SNPs. And you can see in that area where the deletion is that there's only 2 lines in that deleted region, because you have a loss of those heterozygous calls. So basically, you can think of it in a way as like that SNP information is giving you kind of a confirmation of that deletion region, right, that you're seeing on the fluorescence intensity call from the array. Now the middle image, right, is showing a duplication. So this is slightly different. Here we're seeing the opposite. There's an increase in the fluorescence intensity. And then at the bottom, you see an additional line because now you have AAA, AAB, BBA, and BBB calls in this region. So again, kind of confirming that you're seeing that duplication by fluorescence intensity. Now, the bottom image is showing a copy neutral loss of heterozygosity. So there's no change in the chromosomal copy number. You still have 2 copies, right? But you have this region of homozygosity. And this, as I mentioned before, you can see during, if there's uniparental disomy going on or consanguinity in the family. Next slide, please. So now let's shift gears and talk about next generation sequencing. So there's 3 main types of clinical tests that use this technology in the clinical labs. There's the multi-gene panels, right, which is going to test for the most common genes associated with a condition and can range from a few genes to several hundred for really large panels. You have exome sequencing, which is analyzing all of the coding regions or exons in an individual's genome, and then genome sequencing, which is going to analyze all of the coding and non-coding regions of the genome. So the first 2 methods use a capture technology so that we're really only sequencing the regions of interest, whereas with genome, you're sequencing everything, basically, so no capture is needed. Next slide. So in this slide, I'm showing a general overview of a targeted next generation sequencing approach from sample to variants. So again, this would be the case of like a large multi-gene or a multi-gene panel or exome sequencing. But essentially, we're isolating the genomic DNA from the patient's sample, right? And we're going to fragment that DNA into smaller pieces that are needed for the sequencers that we're using, right? And the most utilized technology right now in the clinical labs is a short read sequencing. So these fragments are about 150 to 300 base pairs in length. And so next what we need to do once we have those fragments is to get from a genome's worth of DNA down to just those targeted regions of interest that we want to sequence, right? And this process is kind of what we call part of the library prep and target enrichment process. And so what we do next with those DNA fragments is add universal adapters and molecular barcodes. So molecular barcodes, just think of like shopping at Target, right? Or going to the grocery store. Every item in that store has its own unique barcode, and we can do something similar, right, with DNA, where we have a unique molecular sequence of DNA that we're adding on to that patient's samples so that we can identify that unique patient. And this allows us to then basically multiplex many patient samples together and run them all together on the same flow cell. That helps us be more efficient. It helps us do a higher volume, and especially in a large scale clinical lab. Right now, so the most common technique that we use in the clinical labs to pull out just these regions of interest is called hybridization capture, or you may hear it called Hive capture. But it's, the way I think of it is essentially like fishing, right? You have your pool of DNA fragments, right? And you're adding baits in there. And those baits are going to hybridize to your regions of interest. And you could think of those DNA fragments that you're interested in as like your fish, right? And so then what you can do is basically just pull out those baits and wash away the rest of the DNA that you don't need. And so once you have then just your pool of your regions of interest, you can load those DNA fragments onto the flow cell, onto the sequencer, and then there's a round of amplification that happens on the sequencer. Then from there, what we do is called paired end sequencing, because we're sequencing each molecule of DNA from each direction. And so once the sequencing is complete, then the next step is going to be the bioinformatics process, which is essentially like, how do we go from those raw sequencing reads to the high quality variant calls that we're going to be analyzing further. And so next slide, please. Genome sequencing is different from a targeted sequencing approach, right? Because and most of the differences occurring in like the library prep step, and you don't have target enrichments because we want to sequence the entire genome, right? So basically we're just going to fragment the DNA, add adapters and those molecular barcodes so we can still do some multiplexing of samples together on a single run and then we're going to put them on the sequencer. Next slide. So I just want to spend a moment on two concepts that are important for next generation sequencing, and that is the depth of coverage, which is really a measure of how many times you're sequencing over a given region. And now you may have heard of terms like 20x coverage, right? That means for then that given region you've sequenced over it 20 times. And you may hear of other terms similar like average coverage or minimum coverage, right? Now this depth of coverage is different from the breadth of sequencing coverage, which is really looking at how much of the exome or the genome was covered in sequencing. So exome tests will often say that, for example, like 98% of the exome was covered at 20x. And that's giving you really an idea of that whole breadth of coverage as well as depth. Now, panels typically have the highest average depth of coverage of the three technologies, around 200 to 300x typically. Exomes, it's usually around like 100, 150x. But it's important to remember that this is the average depth of coverage. It's the average, right? So this is going to be variable across all of those regions that are being sequenced in this technology, largely due to, just some of that variability that gets brought in with the target enrichment process. So for genomes, typically you'll see like a minimum 30x coverage for a clinical genome sequencing. The averages can be a little bit higher. But one of the things that's nice about genome sequencing is that your coverage across the genome is much more even. So you really don't need as high of an average depth of coverage when you're sequencing across the genome because you really have this nice even coverage. Next slide. So let's take a look at how read depth is playing a role in CNV detection with next generation sequencing. Now, there's a few different styles of bioinformatic algorithms that can call copy number variants from NGS, but the read depth caller is really the major one that clinical labs are using to call exon level CNV or copy number variant events. So how this works is basically the read depth or the number of times that, again, that you're sequencing over that exon in your patient is compared to the read depth in a control. Now, there's a few different ways to do this that I've seen. Some labs may compare that patient's average read depth at the exon to a pool of normals, the same read depth at that same exon in a pool of normals that have been run, and then maybe those get that pool of normals gets updated at a periodic basis. Or alternatively, you can even take that read depth over the exon in your patient and compare it to all of the other individuals that have been run in that same flow cell, right? Because you're not expecting that at that same exon that you're going to have a lot of other individuals that are multiplexed together will have a same CNV call at that exon. One other technique that I've seen used too is that for genome sequencing, right, you can really take advantage of the fact that you have such even coverage across the genome. So you can compare the read depth of a given exon in that patient, right, to the average read depth across other parts of the genome in that same patient. So in the figure here, I'm showing a deletion in the bottom left, where you have the reduced read depth across that exon. So you can see that's deleted, right? It looks similar, remember, to how when we were looking at those plots from the arrays earlier, right? And then in the next image, right, where the duplication is, you can see how you have that like increased read depth coverage compared, that's been normalized in that duplicated region. Now, the minimum CNV resolution tends to be down to 1 to 2 exon for panels, could get down to 3 or 4 exon size resolution in exome sequencing. And in genome sequencing, we typically say like 1 kb, right? Because it's different in genome. We're looking at the coding regions and the non-coding regions, but it's similar to like what we see in exome sequencing. Right now, remember, right, we're comparing that. If you think about microarrays, right, those events are much, much larger that you're picking up on a microarray, right? You can get down to about 25 kb for a deletion. So you're going to largely really be missing those small CNV events that are smaller than a full gene size when you're doing an array that you can detect on an NGS technology. Next slide, please. So here's a study that we had presented looking at a cohort of almost 12,000 individuals that had both microarray and an exome sequencing performed. And from this analysis, we saw that about 98% or 98.7% of the cases either had equivalent results, meaning that whatever was detected on the array was also detected on the exome sequencing, or the patients had additional findings on exome sequencing that weren't picked up on the chromosomal microarray. Because remember, we could get that finer resolution CNV analysis on exome. Next slide, please. So now let's take a look at all of the different kind of variant types that are covered with each of these testing modalities that I've just gone over. You can see with short read genome that you're able to get almost all of the relevant variant types except for methylation status. Now for exome sequencing, again, you're getting a majority of the different variant types, but methylation status and repeat expansion disorders aren't detected. And you're going to have like a much more limited ability to detect very large structural variants or what we call kind of like those large scale cytogenomic events. You can see here with multi-gene panels, in addition to the limitations that I just mentioned for exome, you also typically don't get the mitochondrial genome with it or uniparental disomy or regions of homozygosity. And with microarray, right, as we discussed, you can get a uniparental disomy, regions of homozygosity, these large cytogenomic structural variants, CNVs, and sometimes you have some sensitivity to detect some of the mosaic CNVs as well. Next slide, please. So let's look at some of the other differentiating features of these technologies. Now, studies have shown that the diagnostic yield on genome sequencing is the highest of any of these technologies, with microarray and multi-gene panels tending to be the lowest. However, there is the caveat that there's going to be a little bit of variability in these diagnostic yields depending on really what your clinical indication is for testing. Also here, you can see that the VUS rates are lower for genome and exome compared to multi-gene panels. And this is largely due to the difference in the reporting styles between panels and exome and genome sequencing, right? Because with panels, typically all variants of uncertain significance are going to be reported because that's just the style of reporting on those on the multi-gene panels, right? So this is really highly dependent on the number of genes that are sequenced. The more genes you sequence, the more chance you have to find these rare variants of uncertain significance. So it's not really uncommon for a large 300 gene plus panel to have 4 or more variants of significance on it, which really may or may not fit with the patient's presentation. Now in exome and genome sequencing, on the other hand, right, as Maria is going to go to in much more detail than me, only those highly suspicious variants of uncertain significance that are fitting the patient's presentations would be reported. So essentially, you could think of these of the ones that are getting reported, they're all going to be, if there is any report, it's going to be a hot variant of uncertain significance. The other thing I want to mention too is that with exome and genome sequencing, these essentially can stay up to date with the literature, right? Since you already are sequencing all of these genes. Whereas with multi-gene panels, it takes development time and work to add new genes to the panel. So unless the lab is really updating the multi-gene panels very frequently, these can quickly become out of date, especially in a clinical area that has a high rate of new gene disease discovery. So lastly, the advantages you get from exome genome sequencing is the ability to run the parents simultaneously with the proband, which we call trio analysis. Also the ability to do reanalysis and the ability to get secondary findings. And so Maria is going to dig into some of these concepts in more detail. And so with that, I will turn it over to Maria.
Hi, everyone. I will take it from here as we go deeper into what exome and genome sequencing is. So let's go to the next slide. So both exome and genome fit under the umbrella of next generation sequencing. And at this time, most clinically available tests use short reads. The workflows in the lab can be broken into the 4 groups that you can see in this slide. However, the order in which they occur could vary from lab to lab and sometimes even occur simultaneously to expedite turnaround times. So the wet lab. The wet lab component includes DNA extraction, generation of the next-gen sequencing data, and depending on the outcome of the case, orthogonal confirmation of selected variants. Then we have the bioinformatics pipelines, which align the sequences from the individual being tested to the reference exome or genome, annotate the variants, which is the process of determining if the change results in a missense or a nonsense variant, for example, and then using those genomic coordinates to allocate frequency in population databases, presence in ClinVar or AGMD, or known classifications in labs' internal databases. Finally, the filtering of variants that do not meet predetermined conditions based on those annotations. You can imagine, for example, excluding variants that are present in over 1% or 5% of individuals in NOMAD, or excluding synonymous variants from some searches. Then we have clinical analysis. Clinical analysis refers to all the steps that consider the individual clinical information. These are usually at least supervised by trained analysts that have different roles, and they include abstracting, variant classification, and variant vetting, which use specific criteria recommended by ACMG guidelines, and we will go over these ones in a little more detail coming up. And finally, reporting, which involves selecting which of the variants present in the proband fit the phenotype and are likely to explain at least part of their clinical features. Then report writing, and finally sign out, which is the final step, and it's performed by at least one board-certified geneticist. Next slide, please. So let's go a little deeper into these steps, and let's start with abstracting. Abstracting is using the test requisition forms, medical records, previous lab and imaging results, any family history submitted by the provider, and a clinical abstractor will take that information and will summarize the list of relevant features. Then those features are translated into HPO terms. And as you can imagine, this is where it's very important to receive the patient's main clinical features as well as any unique or differentiating findings, because that is information that we're going to use for our reporting. Next slide, please. What is HPO? HPO stands for Human Phenotype Ontology. It was first published in 2008, and it currently contains over 16,000 terms. It provides a standardized structure and control vocabulary of phenotypic features that are encountered in human disease. This framework allows clinical data to be standardized for research and clinical applications. So if you look on the right-hand side of your screen, you can see an example of the HPO hierarchy with abnormality of the musculoskeletal system on the top. You can appreciate how it branches by anatomical distribution, and sometimes the branches meet again in the lower tiers to create very specific child terms. Each term at each level can be associated with different diseases or gene disease associations in databases like OMIM or OrphanNet. Next slide. The next step in clinical analysis is variant vetting and classification. So variant vetting is the process of reviewing all evidence for or against pathogenicity at the variant level. These includes reviewing population data, in silico predictors or computational data, functional studies, segregation within a family, and the clinical fit. The evidence can come from publications or the individuals tested previously or in the current case. And finally, ACMG provides guidelines that weigh different types of evidence, leading to a final classification that can go from benign in the one end all the way to pathogenic in the other end. You will notice here that I broke variants of uncertain significance into two different categories. We can have variants in genes with validated gene disease associations, but at this time have insufficient evidence to be sorted as pathogenic or benign. And we have others that are strong molecularly, imagine a de novo loss of function variant, for example, but in a gene with an association that is still emerging or has limited evidence. Those gene disease associations are considered candidate or sometimes referred as GUS, genes of uncertain significance. And per ACMG guidance, variants in those genes cannot be classified as higher than a VUS. Next slide. So as you can appreciate at this point, for the variant selection and reporting, we need input from both the genotype in the form of a variance assessment by type, molecular properties, and mode of inheritance, plus the phenotype, which requires a clinical analyst to cross-reference the strongest variants in the patient with clinical information provided for the test and the phenotype associated with pathogenic variants in a given gene. The end product of this review is a report that includes only clinically relevant variants that could impact patient care. Next slide. So let's talk a little bit about reanalysis, the process of reanalysis that Britt was alluding before. In the next slide, we can see that reanalysis broadly refers to two different processes. We have variant reclassification, which focuses on reevaluating previously reported variants in light of new evidence that could have accumulated. It relies on updated medical literature, data sharing, and laboratory database trends. And on the other hand, we can have a full review of the case either by updating the reference data that includes new population frequency, gene disease associations, presence in PubMed or ClinVar, or even taking it a further step back and rerunning the sequencing data through the full pipeline to open the pool of interesting variants to new variant types like repeat expansions or improve CMV detection and annotation. This is a collaborative process that uses updated clinical reports and reclassification processes, and it can be initiated by the clinician or by the laboratory. Next slide. So let's go over a couple of examples together. So this reanalysis case number 1 was first reviewed in 2018. And at that point, we had a 13-month-old with failure to thrive, relative macrocephaly, lip tie with a frenulectomy, non-contributory family history, and a high suspicion for Silver-Russell syndrome with negative methylation testing and a negative exome trio analysis. The same case was reviewed again in 2024. At this time, we knew the proband was 6.5 years old with short stature, poor weight gain, macrocephaly, and dental crowding. But this time, the family history reported the mother was affected with an adult height of 4 feet 11 inches tall with an expected mid-parental height between 5 feet 4 and 5 feet 6 inches. Trio reanalysis this time identified a maternally inherited frameshift variant in PLAG1. Next slide. PLAG1 is associated with Silver-Russell syndrome. It encodes a zinc finger transcription factor that has been shown to affect insulin growth factor 2. IGF-2 is known to be essential for fetal growth and has been implicated in the pathophysiology of Silver-Russell syndrome. Heterozygous loss-of-function variants in this gene have been reported in individuals with Silver-Russell syndrome, either occurring de novo or inherited from an affected parent. Silver-Russell syndrome is characterized by growth restriction, relative macrocephaly, dysmorphic features, and some limb anomalies. The first report of variants in PLAG1 causing disease in humans was published in 2018. The association was confirmed by additional case reports in 2020. In this case, the updated gene disease association as well as the affected status of the mother both resulted in a new interpretation and a diagnosis for this family. Next slide, please. Let's look at another scenario for reanalysis. So initial analysis for this case in 2020. At that time, 4-year-old with developmental delay, short stature, frontal bossing, and maxillary hypoplasia. This was also suspicious for Silver-Russell syndrome, but the proband had a normal methylation study and normal microarray. Exome testing identified a heterozygous variant in TLK1, which is associated with a neurodevelopmental disorder, and the variant was classified as a variant of uncertain significance. The proband also had a copy number gain at 17p13.1, which was also classified as a VUS. Reanalysis in 2024 now provided additional clinical information that included an elevated hemoglobin A1C and fasting glucose. At the time of reanalysis, we identified a pathogenic variant in JCK. Next slide, please. So JCK is associated with maturity onset diabetes of the young, of MODY. It encodes glucokinase, which is an enzyme that regulates glucose metabolism, and heterozygous inactivating variants in JCK cause autosomal dominant MODY. It is associated with mild fasting hyperglycemia, sometimes starting at birth, but not typically associated with diabetes-related complications. The variant identified in this proband had been observed in multiple unrelated patients from different ethnic backgrounds with MODY, published in the literature. It had also functional support, and all of these led to a classification of pathogenic per ACMG criteria. In this case, reanalysis identified a variant likely responsible for the elevated hemoglobin A1C in this proband, and this diagnosis will impact future management and follow-up. Next slide. So, in conclusion, you can see both exome and genome cases may be reanalyzed months to years after the initial report. This reanalysis is a collaborative process between the laboratory and the patient's clinical team. New clinical information can change the variant interpretation or bring completely new genes to the differential diagnosis. Depending on the study, the diagnostic yield of reanalysis can vary between 12 and 18%. And the main factors leading to that new finding at reanalysis are seen in this pie chart. We have new variant level evidence, like prior observations or new classifications in databases like ClinVar. 35% are new gene disease associations, and about 15% come from bioinformatic improvements. Next slide. Now let's switch gears a little bit and let's talk about the other advantage of exome and genome sequencing, the ability to test the parents at the same time as the affected individual. In the following slide, we can see the differences between trio analysis and non-trio. A trio usually includes the proband and both parents. All individuals are sequenced, but the analysis is performed with the proband at the center of the parental, with the parental samples just guiding us to determine segregation. That is, determining if the variants occur de novo or compound heads. Testing the parents also allows us to determine Mendelian errors that can point to uniparental disomy. The testing process starts with sample collection, submitting it to the lab, and starting the wet lab process, as we described a few minutes ago. After the results are returned to the ordering provider, the patient and their family receive post-test counseling to ensure they understand the significance of the results. Now, in the case of non-trio analysis, which can be either proband only or duo testing, and when we test duos, it can include either the proband and one parent, or less frequently, a proband and a sibling who can be affected or unaffected. On the one hand, having an additional individual can help us determine co-segregation of suspicious variants among similarly affected probands. However, the de novo and compound heterozygosity status cannot be confirmed in the absence of both parents. When we do not have both parents to start the process and we have a non-diagnostic result, let's say we have a variant of uncertain significance reported, the post-test counseling session will need to address the need to submit parental samples to resolve that VUS, which implies another sample collection and submission, confirmations in the wet lab, revetting of the variant in light of the evidence that we can obtain from segregation, and potentially updated reports and the need for a second visit for counseling after the update. Next slide. We're going to go over a couple of examples to illustrate the difference between trio and non-trio testing. So in this first example, an 8-month-old with enlarged ventricles, simplification of the gray matter, microcephaly, cataracts, abnormal eye movements, mixed tone, seizures, dysmorphic features, a micropenis, and possible developmental delay. The family history is significant for a father with seizures and a maternal grandmother with cataracts. In the next slide, we can see the results of proband-only exome testing. We identified 2 heterozygous variants in MINPP1. One of them was a nonsense variant that could be classified as likely pathogenic, and the other one was a missense variant that was classified as a variant of uncertain significance. This gene, MINPP1, is associated with an autosomal recessive form of pontocerebellar hypoplasia, which can cause brain abnormalities, microcephaly, developmental delay, epilepsy, cataracts, and dysmorphic features. As you can appreciate here, there's a lot of overlap with our patient. This is a really good fit. These are suspicious variants, but we cannot reach the threshold for diagnostic because one of them is still classified as a variant of uncertain significance. Now, in the next slide, we can see what happened when we test the parents. The same sample was run as a trio, and when we did that, we can confirm that those 2 variants that we have identified as interesting are in fact in trans. Each one is inherited from a different parent, and that allows us to apply additional criteria, additional evidence to that missense variant. When that missense variant is classified as likely pathogenic, now we can call these findings diagnostic. Next slide, please. Let's look at another example. In this case, we have a 7-year-old with a history of IUGR, developmental delay, weakness, muscle atrophy, and cataracts. Renal ultrasound shows hydronephrosis. We have a brain MRI with a hypoplastic corpus callosum and pachygyria, and previous genetic testing showing a copy number gain at 15q13.2, q13.3, classified as a variant of uncertain significance. Family history in this case was non-contributory. In this case, the results of probe and exome testing identified 3 different variants that could be linked to the phenotype. We had a heterozygous missense variant in DYN-C1H1, which is associated with an autosomal dominant form of neuropathy with impaired neuronal migration. It has several overlapping features with the presentation of our proband, including cortical malformations, lower extremity weakness and atrophy, and developmental delay. But the variant can only be classified as a variant of uncertain significance in the basis of its rarity and the damaging computational predictors. There's also a heterozygous missense variant in RYR1, which is associated with a form of autosomal dominant and autosomal recessive myopathy, which can overlap weakness and muscle atrophy as described in our proband. This variant was also classified as a VUS. And finally, we had a heterozygous missense variant in GRIA1. GRIA1 is a fully penetrant autosomal dominant disorder associated with a neurodevelopmental delay. This variant was absent in all population controls, and it had benign predictors, but we couldn't really rule out if this variant was de novo, it could be significant for the phenotype. So we reported these three variants of uncertain significance. In the next slide, we can see the result of the segregation when we received the parents for follow-up. As you can see, the variant in DYN-C1H1 was confirmed to be absent in the mother and the father. Being de novo and having a great clinical fit makes these the most likely diagnosis for the patient. After reviewing the classification of the other two variants, we found that RYR1 was inherited from an unaffected parent. Because the dominant form of the myopathy can have reduced penetrance, we really cannot add or subtract any evidence to the classification, and that variant will remain a VUS. In the case of GRIA1, the variant was also present in one of the unaffected parents. In this case, because we know that the disorder is fully penetrant and pathogenic variants always occur de novo, we can rule this out as a cause of the phenotype in this proband, and that variant can be downgraded to likely benign. As you can see here, if we would have had the parental segregation at the time of the initial analysis, not only we would have had a diagnostic finding from the start, but we would have reported less variants of uncertain significance because we would have been able to tease them out at the first pass. Next slide, please. So what is the clinical impact of trio testing? I hope that at this point it is clear to you that trio testing is most likely to identify a genetic diagnosis in a patient when compared to patient testing only. The different studies that we have in the table here are performed between 2014 and 2017, so they're not the most up to date, but the trend clearly persists. And we can see that we have a higher likelihood of a diagnosis when we have the parents for testing. If you move to the next slide, we can see that this is especially important when we're looking at individuals that are underrepresented in large research and reference databases. So in a more recent study presented by Jessica Chong et al. At ASAG in 2024, we observed that in a cohort of 73,000 individuals, which included 52,120 trios, the diagnostic yield was the same for individuals that were self-reported as non-Hispanic white versus the aggregate of all underrepresented minorities. And we see the same results when we use genetic ancestry instead of self-reporting ethnicity. And we compare individuals with European ancestry to those non-European. And this is because for these individuals, we can use the parents that have a similar genetic background to tease out these variants that could be benign depending on the disease associations. Next slide. So in conclusion, all genomic testing technologies have benefits and limitations. The diagnostic yield and the rate of variants of uncertain significance differ by technology. The process to get from a sample to a result involves various steps, including wet lab processes, bioinformatics, clinical analysis, and reporting. Genetics is a constantly advancing field, with new gene-disease relationships discovered regularly. Exome and genome sequencing results can be reanalyzed to take advantage of scientific progress and increase diagnostic findings. Trio testing, which as we discussed, means including samples from both biological parents or other close relatives, in addition to the patient or proband, increases diagnostic yields, lowers the numbers of variants of unknown significance, and shortens the time it takes to receive a definitive diagnosis. And with that, I'll turn it over to Kristen to start answering questions.
Thank you so much, Britt and Maria, for a fantastic presentation on genomic technologies, including exome and genome sequencing. We're going to go ahead and start the Q&A session, and we're going to start with some of the questions we received during registration. Just as a reminder, if you have a question for either presenter, you can enter it into the question tab of the GoToWebinar panel. And if we have time, we can try and get to some of the live questions. The first question I'll go ahead and direct towards Maria. We had a question come in asking, how often do you update the ACMG secondary findings list?
Okay, so ACMG updates their guidance every couple of years. The last update was published in August 2023, and we know that there may be another one coming up soon. After the guidance is published, labs take a few weeks to update workflows and the documents that are needed for the reports. So it's quickly after after the guidance is published.
Great. Thank you so much, Maria. The next question we had come in, I'll go ahead and direct towards Britt. The question was, what is the utility of whole exome and whole genome testing in the prenatal space, particularly in the setting of a limited phenotype?
Yeah, thanks. This is a great question. Actually, there was a really cool presentation at ACMG just last week from the New York Genome Center, I believe, where they looked at a cohort that they had done with prenatal genome sequencing. And what they had found was that the diagnostic yield, I think, was about 23.5% on prenatal genome versus, kind of the standard of care for prenatal being array and karyotype. The diagnostic yield was a little over 6%. So it's looking, really exciting, I think, in the prenatal genome space. I know internally we've done a study looking at prenatal exomes that we've done. And when we had evaluated at that time, it was like over 1,500 prenatal exomes that have been done. We saw about a 25% diagnostic yield. So I think the take home here too is you can still get a good diagnostic yield even when you have a limited phenotype, right? Because we're also evaluating, as Maria was going through, like, what are the really strong molecular characteristics as well, and looking at the genotypes that we're seeing in all of the genes and the variants that are coming up in these samples. I don't know, Maria, if there's anything you want to add there.
Nope, I agree.
Yeah, awesome. Thanks.
Thank you so much. Definitely an emerging space that's going on there. The next question we had quite a few questions around reanalysis. I'm going to try and kind of lump them all into one question for Maria. So, is reanalysis a good option for older exomes from 2015, let's say 2015 or 2016? We also had some questions come in around, how long should you wait for a reanalysis if there's no change in phenotype?
So I would say is a good option for older reanalysis, yes, with caveats. And it kind of goes into the other part of the second question. So in terms of the likelihood of finding a new answer, a lot of new gene disease associations and updated variant classifications will be available in an 8 to 10 year period. And that could absolutely impact the outcome. However, there also could be other changes in the lab, in the chemistry or new colors being incorporated, and those are not always feasible to layer into the original VCF file. So you should call the lab and ask if you can still order reanalysis, or if you have reached that point where you're probably going to benefit more from resequencing to be able to add all those additional features in addition to the new evidence that builds up. And how often should you get a reanalysis? It really depends. It's hard to know who are going to be the people that have those variants in the new genes as they are coming up. But in generally speaking, if the phenotype has not changed, think about it for those of us who have been in research. Usually takes a year or two to build up evidence, to have a cohort and publish that data. So I think that is a reasonable time to wait before doing a new review of the case.
Fantastic. Thank you, Maria. The next question we received is, I'll go and direct towards Britt. So how much increase in diagnostic yield will a genome give?
Yeah, so this is another great question. Actually, last summer there was a great paper that came out in the New England Journal of Medicine from Monica Wojcik and the Broad, looking at a large cohort of patients that had genome sequencing. I think it was like around 800 patients, somewhere around that, don't quote me. But and in that study, they found that 8% of the patients in that cohort, right, got a diagnosis where the genome sequencing was really needed. I think many of those patients had previous genetic workup, mostly exome sequencing that, was negative findings, but they found really that there was like an increased 8% diagnostic yield that you really needed the genome to find the variants, like large structural variants, other things that aren't easily detected on exome sequencing.
Thank you so much, Brett. I have another question for you that came in, and it's around insurance coverage. So what does insurance coverage look like for whole genome or exome sequencing?
Yeah, this is another great question. So, I think we're in an exciting time, right? Things are really kind of moving rapidly. We've come a long way, even in the last few years. If you think about all of probably many of you on this call are aware of like the biomarker legislation initiatives that have been going on across different states. So now there's actually 32 states with Medicaid coverage for outpatient exome and or genome sequencing, and 14 states that have Medicaid coverage for inpatient rapid genome. And we see across, when we look at commercial insurers and Medicaid as well, that about 80% of covered lives or individuals have coverage for outpatient exome, and about 45%ish of individuals have coverage for outpatient genome, and it's pretty similar for the inpatient rapid testing as well. So I think the coverage is continuing to expand. And yeah, I'll leave it at that.
We also had several questions around segregation studies for VUS resolution, and I'll offer it up to either Britt or Maria, just asking, how beneficial segregation studies are to VUS resolution, and if the parents aren't available, if testing other relatives is helpful, and when to do that.
I can take a first pass. So how is it helpful? Like the parents are going to be the most helpful, and it is all based on the criteria that we are provided by ACMG. So think about it, it's kind of like a point system, and you need to reach a certain threshold to be able to call a variant likely pathogenic. When we do not have segregation at all, you have to rely on the information that you have in front of you, and you have very little points. If you know that a variant is confirmed de novo with both parents sequence, so you are confirming paternity, you're confirming the paternal relationships by doing exome or genome, you know that they are the biological parents. You're also ruling out all other causes because we're taking a look at all 19,000 genes. And if we have that level of evidence, we can apply a strong criteria that are easily bringing that variant from just a VUS to likely pathogenic. If we cannot have the parents at the time of testing, we're only able to apply a moderate criteria because we can test the variant or variants reported in the proband for their presence in their parents, but we cannot rule out paternity or maternity. So we do not know. We know that in the samples that we get, they have or they don't have the variant, but we haven't ruled out the other things. So we are only allowed to use a moderate criteria in that case. And sometimes that is enough to get us to LPAT, some other times is not. If we don't have the parents, we can use more distant relatives or siblings if they're affected or unaffected, and then we are really like looking at the pedigrees and counting segregations to try to add some evidence, but it gets harder.
Yeah, thank you so much. Yeah.
Maybe I'll just add a little bit to that. I think that was great, Maria. I was involved with a couple of really large studies that came out looking at over 1 million individuals that had variants of uncertain significance. And then there was a follow-on study looking at kind of rates and reclassifications. And some of the things that we have found in those is that, right, not surprisingly, like clinical information by far is like one of the largest contributors to helping reclassify variants of uncertain significance. And when we look at like more deeply at that clinical information, we could see statistically that like having de novo, like being able to tell that a variant is de novo is by far like the most enriched thing we see, that in variants of uncertain significance that get reclassified versus variants of uncertain significance that don't. So it's really important, you know, and I recognize like there's challenges, there's real world challenges sometimes to being able to get both parents, but where you can, like it really can make an impact.
Yeah, absolutely. Thank you so much to both of you. I think that's all the time we have today for questions. Thank you to everyone who submitted their questions. Couple of reminders, don't forget, if you want to obtain CEUs or CECs for today's webinar, just fill out the evaluation by April 3rd. Also, a link to this recording will be emailed out tomorrow to all registrants. Thank you so much for attending the webinar today, and enjoy the rest of your day.